right, continuing on with our therapy development session, I'd like to invite Paula Vij from UC Davis to the podium. So Paula Vij, who is part of Kyle Fink's group, their team received a grant from the STXBP1 Foundation for the work that she is about to present. Thanks, Paula.
Good afternoon, everyone. Thanks for staying in the last session of the day. Before I start my talk, I want to say that I'm an earlier researcher in the STXBP1 field, but I'm very impressed by being here. It's my first time too in the science meeting and the family meeting, and I'm really impressed by the research being done, the community involvement, and it has really motivated me to keep the science forward. So just wanted to say that. Thanks. Like Charlene said, I am Paula Vij. I am a PhD student in Dr. Kyle Fink Lab at UC Davis. And today I want to share our work in another modality or strategy of gene therapy for STXBP1, known as CRISPR activation, or CRISPR-A for short. And so before I dive into what CRISPR-A is, I want to talk a little bit about the background that we've heard already. STXBP1 encodes for a protein known as MUNC18, which is important in the communication of the brain cells. And so this is a picture of a neuron, and if we zoom in the terminal side of the neuron, we find something known as the synapse, and the synapse is where the brain cells, the neurons, communicate with each other. And then we have the presynaptic terminal, which is the cell that is sending the signal, and then the postsynaptic cell that is the one receiving the signal. The signal in the brain cells are in this form of chemical messengers that we know as neurotransmitters, and then they're packaged, or they're in these circles in the presynapse known as neurotransmitter vesicles. So MUNC18 is up there in the presynapse, and its role is to help these vesicles come down to the presynaptic membrane and then being released to have proper communication in the brain cells. Now, if STXBP1 is really important in brain communication and how the function of the cells, what does losing STXBP1 matter? And I think we've heard of this word known as haploinsufficiency. And haploinsufficiency is the condition when having only one working copy of the gene is not enough. So for most of the genes, we have, most people have two copies, one from the parental side, one from the maternal side, and in a normal scenario, they work normally. Now, when there's some type of mutations or the protein, the gene doesn't function, and then there's not enough protein, and so there's no proper brain function. And so this is the haploinsufficiency, and then individuals with this condition and this mechanism develop these different phenotypes that we see. And so it's harder for the cells to communicate, to work properly, and that affects learning, development, and behavior. Now, in the lab, we asked the question, how can we fix that? And so this is another way or another strategy of gene therapy, Cas9, that use another form of Cas9. And so the red or neuron is what happens in a normal scenario. There's a lot of neurotransmitter release. In a disease scenario, very less amount of neurotransmitter release, and in the green cells is more neurotransmitter release. Now, in the middle is this Cas activation system. And if we have an analogy, in a disease scenario, it's like we have what CRISPR-A is doing is helping this knob going from a low expression of the gene to kind of like a high. And so it's creating this effect. So our main question is, can we increase the activity of the healthy copy that it's already inside the cell to help improve brain function? Now, how do we do this in the lab? we work with cells that are derived from patients, and so they're usually obtained through blood works that are really important for us to do research, or through skin biopsies. In the lab, we have the ability to reprogram these cells, and we convert them to another cell type that gives rise to cells of interest that we care to study, in this case, brain cells. So we do develop some type of cells that are called neuroprogenitor cells, and then eventually we differentiate these into neurons, and then we study these cells in a dish in the lab. These are real images from the cells that we work with. So iPSCs is that starting material, then we differentiate them into intermediate neuroprogenitor cells, but we also can differentiate them into neurons. And so what it's nice about working with patient cells is that we have the genetic exact information from individuals that can inform the disease, as well as this allows us to test therapies direct in human brains. Now, once we have these cells, we create them in the lab, we have them in a dish. We want to do some further characterization. We want to understand what's happening at the molecular level in the disease context. So we have 3 different levels of understanding and characterizing these cells. The first one, we ask the question, are the neurons healthier? Do they develop properly? And so using technology, you can see that to your left, there is like some tracing of the neurons that is for us to help us see how the neurons branch, how they connect with each other, how long these branches are. Are they complex? Are they simple? What is the context of these developing neurons? The second question we ask is how the neurons form connections. And that is when we have that synaptic, or that synapse. And so these are, again, pictures of the cells that we have obtained from patients. And we look at the pre-synapse and the post-synapse. We see where they two kind of like are together to kind of measure, are they forming synapses? Are they communicating correctly? And how are they forming these connections? And the final question that we ask and how we look at these cells is how do neurons communicate? We look at electrical activity when the neurons are stimulated. And so we use different technologies. One of them is measuring like the calcium activity in response to a stimulation. That's gonna tell us different things about the function of the neurons, frequency of signals, and the strength of the response. We do this characterization pre-treatment to have a baseline and to understand the disease. Then we deliver the therapy, and we do, again, this kind of characterization to understand the effect of the therapy. Again, this is just our experimental workflow, getting cells from patients, which is really important, understand the disease, create this CRISPR, or your intended technology, in our case, CRISPRa. Once we have a lead potential therapy, we do test this in neurons, and we see the effect, and then we can validate this in further complex system, organoids, animals, etc. All these experiments answering this important question, can CRISPRa safely restore STXBP1? Now, this is, I think, my most text slide, but all I want to say is that we have obtained 4 different cell lines with different variants, and they are representative of different sites in the protein. And so we really want to study the effects of the different variants at the molecular level. Now, now going into CRISPRa, there are 3 main components for this potential therapy. The Cas9 protein, maybe you've heard in the previously or in the talk about CRISPR-Cas9 system, which it's the traditional way is to edit DNA to create some sort of changes in the DNA. But in this CRISPRa technology, we use a dead Cas9 protein, meaning that is not going to cut the DNA. Instead, it's going to be used as a vehicle to carry or to transport things that we can put together with the Cas9 protein. The second thing that is used is called these activators. And the activators are the ones that are kind of going to help doing the work of activating the gene that we are intended to do or to have an effect in. Now, these two things need to go to the desired part of the genome. The genome is quite large, and so in this case, we want this complex to go to the STXBP1 gene. And how do we do this? We do it through the use of known as guide RNAs. And so if you can think of them as addresses, they're going to tell this complex of where part in the genome go. Now, how do we work this system? We design multiple guides. Like I said, there's different regions in the STXBP1 gene, regulatory regions that we can target this technology. We just don't know which is the best region to create the best effect. And that's the reason why we screen multiple guides to see which one is the most effective. We designed this and we create this in the lab, and then we test this into patient-derived cells. And then once we see that we have a lead candidate that is creating a good effect, then we select that as our potential therapy candidate, and then we do further validation. Now, Dr. Davidson, she gave a really good introduction and talk about AAV and how is this the delivery technology currently out there for gene therapy. Now, one of the limitations is that it has a very size capacity. So for the CRISPRa technology, when we combine it with these activators, with the guides, it becomes pretty big. And so it doesn't fit into one single AAV. And so in the lab, we're exploring different technologies, different systems for CRISPRa system. One of them is that we can divide this system into 2 vectors, and then we can deliver these to the cells and have a more potent effect. Now, the challenge with this is that both vectors have to go into the cell, and this is quite challenging. Another option is to have another version of the Cas9 protein, which is smaller, can fit into one AAV, and can be easier for delivery. So we're exploring these ideas and that can potentially be of use for the CRISPRa technology. Our overall goal is to find the best balance between the size, safety, and effectiveness to increase STXBP1 expression. Now, when we test these 2 systems in the lab, what we've seen is that the system that we can split into 2, we found a potential lead guide that has the ability to upregulate the expression of this gene. So the gray bar is the base level expression of a patient cell line of STXBP1. When we deliver this CRISPRa technology, we see that expression it's increased. And so we are in the process of further validating this, seeing if this produces, again, the protein that is needed to rescue these brain function deficits that we see. When we tested the mini Cas9 system, we didn't see quite of the same effect. So we're currently optimizing this, trying different guides, trying different effectors that we can test in this. So yeah, we're currently working in this optimization. Now, why do we believe this approach can work? There's another disorder that I work in collaboration with another lab known as Pitt-Hopkins syndrome. It's also a haploinsufficiency disorder, and the goal was to implement the same technology, CRISPRa, and to see if we can restore neuron function. When we delivered this construct to the patient-derived neurons, we saw a partially rescue of some of the neuronal deficits in the neurons. So this is exciting because it's, again, another haploinsufficiency disorder like STXBP1, so we believe this can be of use as a potential therapy for STXBP1. So in conclusion, we have successfully increased the STXBP1 expression using one of the CRISPRa system. We have identified potential lead guide that we're currently doing more testing, more validation, and more characterization. And then we next would determine if this guide in this system is good to restore neuronal function. And with that, I'll just thank the lab, the foundation once again for having me here, giving the talk and allowing me to learn, our collaborators, and I'll take any questions.
Any questions for Paula? Oh.
Hi, thank you. Just two quick questions. One, when you said patients and the last slide, and I didn't catch the disorder, but do you mean human? And two, what did you hear today that you think could be collaborative with the work that you're seeing? You mentioned it a few times, and I'm just trying to understand how you're seeing what you heard here or at the science conference that you feel you can take back that could be exciting.
Yes, for the first question, yeah, it's patient-derived cells that we work with, human patient-derived cells. And for the second question, I've talked to other scientists in the meeting that they have work in the system too, and they have insights. They also have experience doing other disorders, and so they have given me insights in how we can kind of like not follow same trajectories that have not worked for them. That way we don't follow in the same like fail path, but kind of like other advice to look into other things, optimize other things that can accelerate this. So more so in the research, and then here all the, not particular for me, for my research, but hearing all the history studies and all these things is really encouraging to see that not just at the molecular level research happening, but in all the areas that it's advanced in the field.
Can you talk a little bit about the delivery mechanism for a CRISPR gene therapy?
For in the lab or, yeah?
In the lab, but I guess ultimately, if we were to use this in patients.
Yes,
Maybe
in the
just
lab
to
we
add to that, like I think that explaining to people that the dual vector versus the mini one, what really is actually the benefit of having one versus
Over
two,
the
except beyond efficacy.
Right. In the lab, we have different delivery methods that we can do in the cells. We do lentivirus, AAV virus, we do plasmid delivery. Again, those are just for experimental scenarios. If we want to think about translating that to potential therapies, the only way we can currently do that is using AAV for gene therapy. And so it will be, again, same gene therapy for potential clinical translation. For gene, same as gene replacement, it will be for CRISPR-A.
So we need to put your insides into our capsids.
Yeah.
A collaboration made at the summit.
Should I? Okay, I was just going to answer to Charlene's point of what's the difference between the two vectors versus one vector is the beyond efficacy is that, yeah, pretty much efficacy that two can have, it's more amenable. We can work with it in different ways better to create a more potent effect versus just having one that is the mini. So we're currently just have looked at efficacy of the two systems and how will that translate to the clinic? Yeah, we're still optimizing and understanding both of them, yeah. Again, there's, as more research is done, there's other versions of the Cas9 protein that, again, talking in with one of the scientists here gave me an insight in another version that may work better than the smaller compact Cas9. So, yeah. Yeah.
Thank you.
Thank you.